First, I just wanted to say thank you for waking up early. Everyone on the screen except me has had an alarm in the fives, and that's a big ask, so we really appreciate it. Thank you. Um, Virginie, you're on the same time zone, right, as everyone else here?
Yeah.
Correct? Yes, good.
Correct.
I'll just quickly introduce myself, and then what I'll do is I'll go through a few slides, and then I'll let you CAMP4 take over. Does that sound okay?
Sounds great.
Yep. Or you could introduce yourselves as well. Let's just do that at
Yeah,
the
why don't
top.
we do that? We did that
Yeah,
yesterday. That worked. That was
perfect.
easier.
So, Yuri, why don't you kick it off on the CAMP4 side?
Okay.
You ready?
Yep, I'm ready.
As in, just introduce yourself for everyone.
Okay. Greetings, Yuri Maricich. I'm the Chief Medical Officer at CAMP4.
Thanks,
Hi,
Yuri.
everyone. I'm Caleb Moore. I'm the Chief Business Operations Officer at CAMP4.
Thanks, Caleb.
Hi, I'm Dan Tardiff. I lead the discovery team at CAMP4.
Great. And Virginie, who are you? What do you do?
Hi, I'm Virginia McNamer, and I'm the president of SRF.
Thanks, Virginie. For those who don't know, I'm Danielle, and I'm with SRF Australia. So I'm going to take you through some slides, everyone. We've got two QR codes on the first slide. You can see here CAMP4 have pulled together a few key terms that they're going to go through in their presentation that you might not necessarily know. So you might want to just scan the QR code and find out what those terms are. And then the bottom QR code is just our generic SRFA glossary of terms, so just generic terms that you might find there. Okay, we're going to talk about clinical trials today. There we go. Okay, so why participate in a clinical trial? I'm just going to talk for as little time as I possibly can, just to frame and give some context as to what CAMP4 are here to talk about, and then I'm going to hand over to CAMP4 because that's really who you want to hear from. So why participate in a clinical trial? So as you know, most people know I've got two kids with Syngap. So for me, the first point on this page was something that I've thought seriously about, and that is we're not comparing participating and the risks that it might have to no risk in not participating. No risk, not participating does not equal zero risk for us. So this is something that I've really had to frame for myself. One sec, I just want to make sure that everyone getting messages. Okay, everyone can hear, I think now. You can't be seen, guys, but we hopefully you can hear. If you can't hear, then you need to change some settings. You can also type into the Q&A at any point, and we'll get to the questions in a bit. Okay, so yeah, for me, not participating does not equal zero risk. That's a big one for me. And then the other big one for me is we're fortunate to have the opportunity. Most people don't get the opportunity of having a precision medicine treatment, let alone in their city or in their country. And then the other reassuring part about participating in a clinical trial is once it gets to humans, it's actually gone through rigorous safety testing. So that's reassuring for me as a parent. You can see here other potential benefits, fastest way to get access to disease-modifying treatments. ASOs actually flush through the system in 2 or 3 months. The team at CAMP4 can talk a bit more about that, but that's actually allows some flexibility, and it's less permanent for us when we're thinking about our family members. You also have free access to the new treatment, otherwise unavailable, and you'll also get some medical care during the trial. And altruistically, you'll help advance knowledge and treatments for Syngap. So that's great as well. So you can see the potential risks are it might be uncomfortable, you might need to travel, you could get the placebo, and the treatment might not work. So they're some of the potential risks in participating as well. Okay. Oh, there we go. Okay, so I've a bit of a disclosure here. I have adapted Mike's SRF slides, so I've tried to Australianize them, but you'll see the ones that I've Australianized and the ones that I haven't. But this one, the Clinical Development 101. So what you need to know about clinical development. So what you should know is that any medication must be approved by government agencies, and in Australia that's the TGA. So you can see the American, the European ones, and the Australian one is the TGA. Historically, it's been much harder to get treatments and challenging to get treatments to humans. And that's why we have this randomized placebo-controlled trials, which have now become the gold standard because of the placebo effect. So the CAMP4 team can talk a bit more about this, but there's a stat, something like 30% improvements in some studies have been based on just placebo because people thought they were getting better, or the people who were measuring it said that the hope was real and it was coming through as it wasn't real improvements. It was just the hope that was skewing it. So that's why they do need to do these randomized placebo-controlled trials to have those the placebo group as well. I mentioned before, it's gone through rigorous testing, and that testing in most cases, and definitely with CAMP4, is that it's tested in monkeys and mice. Okay, so typical clinical development, you can see there's 3 phases and then it might get approved, but with rare disease, there are some differences. And what you can see here is that phases 1 and 2 are generally merged. And 2 and 3 could also be merged. We'll see if the CAMP4 guys might take us through this as well. But you can see the phases 1 and 2 will be a very low dose, and it's just for safety. You're looking at safety initially, and then they'll ramp up possibly. And then you can see OLE, which is open label extension. So that's when the patients who have the very low dose, so it's most likely not therapeutic, they will get access to the open label, the drug on the open label extension. Okay, so I don't want to harbour on this too much. I just wanted to really call out the time to treatment for some of these drug deliveries or drug discoveries. So you can see preclinical research, that's what CAMP4 have done to date. It's 1 to 2 years. Clinical trials, hopefully we're going to start, and that's something that CAMP4 won't tell us when, but hopefully soon, but that'll take 1 to 2 years. And then TGA approval in Australia can take 6 to 12 months. So if you're not in the trial, you're looking at possibly 4 years before you get access to the medication. Then there's various special programs for rare diseases approved to be fast-tracked, their approval to be fast-tracked in Australia. So you can see here that rare disease ASO trials are not new for DEEs. We had our conference, which Yuri came to a month ago, I think it was now. What are we at? 5 weeks ago, the GETA conference, and we had Andreas Brunklaus talk to the Drave trial. They're in phase 3, and I've actually QR coded the YouTube clip because I watched those videos of the little girl that he was treating in Edinburgh. I think it was Edinburgh. Sorry, it's Glasgow. It was Glasgow. He was treating them in Glasgow, and the little girl showed videos of the little girl before and after having the ASO treatment for Drave, and honestly, I cried just watching it. And I know there's people on the call who there's 28 people on the call now, but I do know that there were people in the room there with Syngap loved ones who also found it quite moving to see the change in functionality for this little girl. And that was the case for most of the people. But you can also see there's lots of other trials for DEEs that are going on. None of the treatments have been approved as of yet, I think. The CAMP4 team can possibly talk to that in a bit more detail. Okay, so what I wanted to mention is there are some things that the CAMP4 team won't be able to answer today. They will be able to answer it, but not necessarily today, and that's for various reasons. Predominantly it's because they're regulated. There's financial regulators because they're on the stock exchange, and medical agencies. So they've got to be really careful about what they can and can't say at this stage. So what they won't be able to talk about today is where the study will take place, the eligibility criteria, the timing, and the costs once approved. There might be a few other things I've missed, but they were the main categories that I really wanted to know that they won't be able to tell us today. And then we all have to be careful when we're participating in these trials. So this is just a reminder that we all have to be really careful about what we say because we're being watched by communities. Okay, now what I wanted to do is hand over to the CAMP4 team who are going to go through their presentation. Caleb,
Great.
is that
Thanks,
you?
Danielle. Give me 1 second and
I'll
I
stop
will pull
sharing,
up a
shall
couple
I?
of slides.
Yep. And I'll have a look at the Q&A. I just wanted to remind everyone the Q&A, I'll be monitoring it. So let me just pop some questions in there if you have.
Danielle, can you see my slides okay?
I can. Yeah, they look good.
Perfect. Great. Well, greetings to the Australian SRF community, and Danielle, thanks for the very kind introduction. I have to say we really appreciate the opportunity to partner and work with SRF Australia, and just for everybody who's participating in the webinar, I have to tell you our relationship with our SRF goes back a little bit. In fact, is sort of foundational to this program and where it is today. We first met Mike Graglia at AES in 2022. It was in Nashville, where we presented our first poster describing our very initial work in Syngap. And Mike was one of the attendees at the poster session and came screaming up to Ali, who was presenting the data, and said, "I have the iPSCs and I need to get them in your hands, and you guys need to be testing them." And that was a real catalyst for us in driving this program from what really is a research project to a full-blown program that's on a fast track to the clinic. So we've had a great collaboration and partnership with SRF historically, and we're really thrilled and honored to partner with you and the broader community in this way, more in the form of education, and we look forward to lots of more opportunities going forward. Danielle, thanks for the appropriate previews on us as a company, as a public company. Just very quickly, we're going to make myself and my colleagues, we're going to make some forward-looking statements. Please bear with us as we move forward through those as a public company. I thought it'd be helpful just to ground everybody and give you some context for who is CAMP4. And our company is really one of our goal is we want to be pioneering a new class of medicines to increase gene expression. And more specifically, we are pioneering the discovery and development of regulatory RNA targeting antisense oligonucleotide or ASO-based therapies to upregulate gene expression and restore healthy protein levels. Now that is a mouthful, and our goal in preparing for this webinar was really to help distill and simplify that in a way that it becomes much more understandable in terms of what we're doing and how our medicine works. So that's the goal for today. And I really want to drive home the point that CAMP4's special sauce, if you will, is our understanding of the regulatory RNAs that our medicines target and the role that they play in gene expression. Simply put, our goal is to increase Syngap expression or make more Syngap protein. So Yuri and Dan and I are speaking on behalf of the folks that you see pictured here. We're roughly 45 employees. We're headquartered in Kendall Square in Cambridge, Massachusetts. Please look us up if you're ever stateside. And the founding science for our company really comes from the Whitehead Institute, Boston Children's Hospital, and Harvard Medical School. So we figured we'd start with ASO therapy for Syngap patients and really defining what is an ASO. First off, some terminology. ASO stands for antisense oligonucleotide. And there are lots of terms that are used interchangeably. So people talk about ASOs, people spell it out, ASOs, oligos, nucleic acid therapies, all the same thing. And these are small nucleic acid molecules that can be engineered to target specific sequences of RNA. ASOs can fine-tune genetic instructions, helping to regulate production of a specific protein. And in our case, we're developing an ASO that aims to increase expression of the SYNGAP1 gene to near normal levels, improve how neurons transmit signals, and alleviate the symptoms of Syngap1 patients. We're using very standard ASO chemistry, very similar to that of Spinraza, which is a drug or medicine that's been approved for spinal muscular atrophy, SMA, by a company called Biogen. And I mention this because I want to highlight that the chemical backbone of our medicine is nothing new. It's been tried and tested. And so we like to say that we're really pointing a known tool at a new target. Coming back to this idea that our understanding of the regulatory RNAs and the role that they play in gene expression is really the fundamental part of our platform, and again, our special sauce. We also thought it'd be helpful just to situate ASOs in the context of other types of medicines that are typically used to treat CNS disorders. So from left to right on the slide here, we've highlighted small molecules, gene therapy, and ASOs. If we focus on small molecules for a second, again, these are the medicines that most people are familiar with. They're small compounds, typically pills. They're taken orally, so take them by mouth. They're absorbed in the gut, into the bloodstream, and travel throughout the body, where they can be absorbed. They get processed by the liver and they get excreted. These small molecules are typically, as I mentioned, they're excreted, so you have to take them repeatedly, typically daily. And the function that they serve is often to bind receptors or proteins or impact signaling pathways in the body in some way to elicit an effect. I move to the middle column, gene therapy. So gene therapy is taking a synthetic version of the damaged gene and packaging it inside of a virus or a viral vector. And gene therapy is shuttled directly to the brain, and it's intended to be a one-time therapy. The reason it's one time is because is a good reason. Our bodies are really good at the human body can identify viruses, and our immune system is very good at responding to those. And so you get one shot with a gene therapy, and it's got to be the dose can't be adjusted and the medicine can't be washed out, and it's very much intended to be a permanent therapy. And then if we focus on ASOs, again, these are nucleic acid molecules that are intended to interact with RNA in a very specific manner. They're administered through intrathecal injection, which we'll spend a little bit more time making sure everybody understands. But we want to make sure that our ASO medicine is able to reach the cells that matter in Syngap. So it's injected into the spinal cord and travels directly to the brain. And this is intrathecal injection. Typically, other ASOs that have been administered for CNS conditions are administered on a monthly or quarterly basis. And we're still working through that process to how ours will be administered. And very importantly, the dose can be adjusted or even stopped depending on the effects that we see. So we like to say that the dose can be titrated up to see the efficacy that you want as we see how patients respond to the therapy. This slide we created just to kind of communicate the point that ASOs are nothing new, and specifically ASOs for CNS conditions. They've been around for a while, and the benefits of ASOs have been shown with multiple approved medicines. If you focus first on the left-hand panel, this is a very simple schematic of sort of the central dogma of molecular biology, whereby DNA makes RNA, makes protein. And ASOs can target pre-mRNA or mRNA in a very specific way to elicit effects on gene expression and protein production. In our case, again, we're working at the first part of the process here and targeting regulatory RNAs to increase the expression of Syngap1. The table on the right-hand side of the slide illustrates several of the approved antisense oligonucleotide or ASO therapies, and you can see that it's across a range of different types of diseases. And we also want to highlight the different ways that these ASOs are delivered, depending on the tissue that is sort of target for the therapy. You can see that approved ASOs for CNS diseases are also delivered by intrathecal injection. And in fact, more than 14,000 patients have been treated with Biogen Spinraza for spinal muscular atrophy, also delivered via intrathecal delivery. So I'm going to turn it over to my colleague Yuri to talk a little bit more about intrathecal delivery and our mechanism of action.
Great. Thanks, Caleb. And also thank you to Danielle and to the SRF and families in Australia. So you all were very kind to host me several times and have shared much about your stories. And those have just both been incredibly motivating, but also very educational to help us better focus on what we're all collectively trying to accomplish here. So as Caleb mentioned, intrathecal delivery of ASOs in order to reach the brain is not something new. It's actually becoming quite commonplace. In this approach, a patient, so potentially a Syngap patient, would be administered via a intrathecal injection. Some people refer to this as a lumbar puncture or spinal tap. And so the IT is then injected directly into the space that then bathes the fluid that goes throughout the lumbar spine as well as around the brain. And this is actually a very effective way to deliver a medicine with the appropriate dose to the regions of the brain that we care about in Syngap and in other diseases. So it allows the ASO to reach the target cells in the brain, but it also has some benefits from a safety perspective, because what we're doing is we're concentrating the medication to focus on those areas that are important and limit its ability to get at high doses to other organs, such as, say, the heart or the liver. And so you can think of it as a very targeted or precision approach. If we were to deliver an ASO systemically, and that's done for different diseases, we in fact, and I think I've shared, have another program where it's done systemically, but that one has a targeting molecule to take it directly to the liver. The challenge is crossing the blood-brain barrier, because our brains have a strong protective mechanism. And so that makes it challenging to get the right dose level or concentration to those areas. And I think it's really important to underscore what Caleb mentioned, which is that Spinraza, as just an example, this is a medication for children with spinal muscular atrophy, and it has been quite common now. So over 14,000 administered patients for that. And so many neurologists and pediatric epileptologists are becoming quite familiar with doing this. And so there's a lot of experience and expertise that we have developed over time. If we go to the next slide, I think just want to highlight too that because of the success and the safety experience here, this is becoming a more standard approach for delivering multiple medicines. So what you see on this slide here is in fact a number of studies across other pediatric developmental epileptic encephalopathies, of which we consider Syngap one of them. And so you can see there's studies across Rett, across Angelman, across Dravet, across other specific genetic diseases. And so I think this just highlights that this is an effective approach, and it's an approach that we think is optimally suited to the technology. There are obviously innovations that are being worked on about additional ways to potentially eventually deliver different types of genetic medicines to the brain, but this is today what is considered to be safe and effective, and hence we see many more taking this approach. If we go to the next slide, now I think we want to start moving towards the data that we're eager to share with you. And so this is just a bit of a refresher on haploinsufficient diseases, of which Syngap is one. And I think one of the things I also just want to highlight at the beginning, and Caleb described this nicely as well, is that when we think about different technology approaches for potentially targeting different diseases. Some technologies are better suited to different disease conditions. And in particular, we think that the approach that we've been working on is really a natural fit because of its ability to basically increase gene expression to a level that is meaningful. So in haploinsufficient diseases, we all have two healthy copies of our particular genes, in this case, Syngap. We get one from each of our parents. However, in those affected with Syngap, they have a healthy copy and they have a mutated copy. And in many cases, that mutated copy makes no Syngap protein. However, if we go to the next slide, the approach that we're using at CAMP4 is to basically target what Caleb described as the regulatory RNA or regulatory element. So this is part of the machinery that actually increases or halts or decreases gene expression. And we've identified actually a specific regulator that tells the gene to make less of the gene or to hold constant that amount of protein that's made. And it's visualized schematically here to the left, right at the level of DNA. And I think it's important to point out that this is at the level of transcription. So this is the first step here. If you look at other medicines, for example, Spinraza, they act after the step of transcription. They act at a splice modulating. And so that just changes its ability of potentially how much protein might be able to be made. So CAMP4 targets at this, our ASO targets at this stage, at this particular regulatory RNA. And if we go to the next slide, what this actually shows now is how it works. So in individuals who have 50% protein expression, you can see that there's a little repressor labeled to the left. And our ASO actually binds to the repressor, and it causes a change in that repressor so that it actually comes off. And because of that, there is now less or no break on that gene expression. And so we get increased protein. And that allows us to take what was 50% and move it to near normal levels. And I think it's important to note that these additional proteins are coming from the expression of the existing healthy gene. So we're essentially just harnessing that healthy gene to make more protein from the healthy gene. And so with that, what I'm going to do is turn it over to my colleague Dan, who's going to now walk you through some of the data that we recently presented at the American Society of Cell and Gene Therapy, which is one of the largest genetic medicines conferences in the world. So take it away, Dan.
All right, thank you, Yuri, for that introduction. Excited to tell you about some of our data for Syngap that we're really, is providing a lot of confidence in us moving forward. Next slide. So before we get into that, just want to highlight that we are at this point already a clinical stage company. Yuri was referencing a program that we have in the liver in a metabolic disease called urea cycle disorders. And this is a study that's actually in phase 1 study in Australia at this point. But our first CNS program is Syngap. And this at the current stage is approaching toxicology studies, and I'll tell you what that is in a moment. But in addition to Syngap, we also are interested in pointing our technology to diseases as well. So we're continually thinking about, what diseases or what haploinsufficiencies can our ability to increase gene expression have some benefit. So hopefully beyond Syngap, there will be a lot more to come. Next slide. All right, so I'm going to walk you through some data first in cells in a dish, and then I'll talk about mice, and then I'll talk about some non-human primate studies, and I'll try to make sure it's clear, what the impact of these different experiments are. So using our platform, we identified this regulatory RNA that Yuri was discussing, and then we identified through a whole process a candidate sequence, a single oligonucleotide that we're ultimately planning on moving towards the clinic. And one of the studies that we ran fairly early on was in some cell type called a patient iPS-derived neuron. So what we were able to do, and this is really conventional across the industry to look at these types of diseases, is to generate neurons from a patient cell type. So you can take skin cells, essentially a skin biopsy from an individual. You can turn those skin cells into what we call an induced pluripotent stem cell. This is a cell that can differentiate into any number of different cell types. We can turn it into neurons. You could also turn it into cardio cells, or you could turn it into muscle cells. But we can turn this into neurons and then look at the activity of our oligonucleotide. So we had a Syngap patient line that had a mutation that resulted in a 50% decrease in Syngap levels. And we had a sibling control. And you can see that consistent with a haploinsufficiency, there's a 50% lower Syngap level in the patient iPS-derived neuron. And when we apply our oligonucleotide to these cells, we see a twofold increase in Syngap levels, restoring it towards that of wild type or levels, or the levels in the control. So this nicely tells us that the oligonucleotide that is targeting the regulatory RNA, when applied to a patient-derived cell type, can restore Syngap towards wild type levels. Next slide. Now, it's really important that we can evaluate the activity of our oligonucleotides in the context of a fully intact brain and tissue. And to do this, we turned to a mouse model that was generated by Ben Prosser's lab at UPenn in the States, and he's part of, he is running the ENDD Center of Excellence that we've also been working with. And this model is a humanized Syngap mouse. And essentially what that means is a human Syngap gene, including the promoter and the regulatory elements, is actually replacing the mouse gene. So this mouse no longer makes any mouse Syngap. It only has either 2 copies of human Syngap or a single copy of human Syngap. No, there's no mouse Syngap remaining. And this provides an opportunity, one, to see, is there a decrease in Syngap in the when there's only 1 copy of the gene? And you can see that comparing the first 2 bars, the homozygous control where there's 2, and the haploinsufficient in gray where there's a single copy, and there's about a 50 to 60% decrease in Syngap levels. And then this also allows us to apply our oligonucleotide and assess whether we can target the regulatory RNA to increase gene expression. And upon a single administration of our oligonucleotide directly into the brain tissue itself, or into the ventricle within the brain, you can see that there's a 2-fold increase in Syngap protein. And this is approaching that of wild type levels. So this is very important because it tells us that we can target the regulatory RNA in human cells in a dish and increase Syngap, but we can also administer the oligonucleotide to intact animals with a fully functioning and developed brain and target the regulatory RNA to increase gene expression, to increase Syngap protein levels. Next slide. Now we can go beyond just looking at Syngap levels. That's obviously very important, and that's what we're trying to achieve. But we also want to look at what's the impact of this increase. Is it making any difference on these animals? Okay. So in the literature, there's a number of papers with regular Syngap mouse models where there's a deficiency in Syngap, and there's very clear phenotypes in different aspects of behavior. There's motor function deficits, there's spatial learning and memory and cognition, there's activity behaviors or seizure activities. And these have all been shown to be to have a phenotype in these mouse models. But this humanized mouse that I talked about in the prior slide has not been evaluated phenotypically before. So we first set out to make sure that there were the phenotypes. There was a difference between the 2 copy and the 1 copy Syngap mice. And you can see comparing the first bar and the second bar and all of these, the first 3 graphs there, that in a rotarod assay, which is looking at motor function, in this wide maze assay that is looking at spatial learning, memory, cognition, as well as this hyperactivity assay that's sort of tracking the movement of mice over time, you can see that there's a significant difference between the wild type and the haploinsufficient mice. And importantly, when we administer our oligonucleotide, these are restored, and these 3 phenotypes are restored upon treatment with our ASO. And you can see on the right the actual protein that is being made from in these mice after they've received our oligo. So treatment with the oligo is reversing these phenotypes, and it is also increasing Syngap protein to a significant extent. So this is very, great to again see that the increase in Syngap is resulting to a phenotypic benefit in this mouse model that's targeting the human Syngap regulatory RNA. Next slide. All right, so these studies are great. They showed we're having a benefit, that we're increasing Syngap. One thing that you may have picked up on is I mentioned that the oligonucleotide was administered directly into the ventricle and the brain, right? That's not going to be the route of administration clinically. We already, heard from Yuri talking about IT administration, so that's how we plan to administer our oligonucleotide. So it's important for us to use a species that we can do IT administration and confirm that we can get enough oligo to the right brain regions to have an increase in Syngap expression. And this is important because, when you do the injection into lumbar spinal cord space, the ASO has to travel a bit of a distance, right? So it's really important that we can demonstrate that with that route of administration, the ASO does end up where it needs to go. So we performed a study in which we administered 3 different dose levels, a low, medium, and high. We gave each dose 3 times, so with 2 weeks apart. So every 2 weeks, animals received a dose of ASO at the 3 different dose levels. We looked at a few things. We looked at the ASO in plasma. We looked at it in CSF. And then at the end of the study, we sacrificed the animals and looked at the brain. And that's really important that we actually understand what's happening in the brain. And that's why this is a very important study for us. And really, I'll show you both ASO concentrations and protein. Now, the key takeaways from this study, first, Caleb, if you could yep, thanks. First, the study, the ASO was very well tolerated. And I think that this is very important for you to understand, is we pay as much attention to safety as we do that we're increasing Syngap. We don't have a drug if it's not safe. So throughout the development of all of our programs, we really place a premium on safety to reduce that risk so that patients would experience. And the 2 take-home messages beyond that this ASO was very well tolerated was that we got a significant ASO concentration in a very dose-dependent way, meaning at higher doses, we got more ASO there in all the relevant brain regions for Syngap, and that this ASO concentration resulted in a significant increase in Syngap expression. And now I'll walk you through that data. So in this slide, we're looking at the oligonucleotide concentrations that we measured in different brain regions. So, there's a constellation of phenotypes associated with Syngap. So it's really important that we have good broad distribution of the oligonucleotide. That means it's reaching multiple brain areas in an even way, so that we're able to engage or target the regulatory RNA that our oligonucleotide binds to. And it's also important that an oligonucleotide achieves higher concentrations in tissue with increasing dose. And that's what you see here, is that in all the different brain regions that we feel are relevant to Syngap, there's a dose-dependent increase in the amount of oligonucleotide that we were able to quantify. And so this really sets us up to then look at the Syngap protein in these different brain regions. Next slide. And that's what we're showing here is Syngap protein in this case, 4 different brain regions associated with different aspects of Syngap. That there's either phenotypes or, for example, in occipital cortex, there's a EEG signature that's relevant. And what you can see is that there's a dose-dependent increase in Syngap protein in all these different brain regions. We also looked at some additional regions that are not shown here, but really what we found is that if there's oligonucleotide that is reaching a brain region, we're able to measure a dose-dependent and significant increase in Syngap protein. And keep in mind also that these are wild-type animals. They already have 2 copies of Syngap, so we're basically increasing Syngap beyond, but we are demonstrating target engagement. If you look back to the mouse studies, we see approximately a 2-fold increase in the haploinsufficient state. So we're extremely excited by this, that we're getting enough drug to the right brain regions, and that amount of drug we're getting there is increasing Syngap in a meaningful way. So again, just to kind of remind you of what I told you, we were able to identify this regulatory RNA. We were able to then discover the antisense oligonucleotide that most specifically targeted this regulatory RNA to increase gene expression. We then showed data that we can recover Syngap levels in a patient iPS-derived cell type, so a relevant human cell type that is expressing Syngap and functional, and restoring it towards wild type levels. We then showed that in humanized mice that are haploinsufficient for Syngap, that we can target the regulatory RNA and increase Syngap in the context of a brain of a living animal. And that this increase in Syngap that we're able to achieve is reversing relevant phenotypes displayed by this model. And then when we administer to non-human primates by intrathecal administration, which will be the clinical route of administration of our candidate, that we get enough ASO to the right place in the brain, and that this results in an increase in Syngap expression. And importantly, again, I mentioned this, but the ASO has been well tolerated in exploratory safety studies. But really, the next key thing for us in this program is to advance into what is called GLP, but formal toxicology studies required by regulatory agencies to advance a molecule from research and discovery into the clinic. So that's the next step happening later this year. And with that, I'm done with the preclinical work and pass it on to Yuri for the final slide.
Great. Thanks so much, Dan. So maybe just want to underscore a couple key takeaways. I think one is Dan showed, this is, I think for us, what we would oftentimes call robust, or I'll use the more human, like exciting data to show that by actually getting ASO to the regions of the brain that the scientific community thinks is important, that we actually see an increase of protein, and that we see that particular aspects of the Syngap condition. So some of those different tests that Dan walked through actually are corrected back to a normal humanized version of the Syngap mouse. And so what that means is now we are going to start what are a set of formal toxicology studies. As Dan mentioned, we look at all the safety and toxicology as we do these, but there's a specific set of studies called GLP, or good laboratory process toxicology studies, that are used to file with regulators as we prepare to go into potential clinical trials. And then the second thing is that we are engaging, as we already have and are going to continue to do so, with your community, patients and families, as well as scientific and clinical experts worldwide, to really start to determine what exactly will the study and studies particularly look like. And this will also allow us to kick off starting to engage with regulatory authorities and clinical sites at the appropriate time. So as Danielle mentioned, while we're not yet in a position to give specific details of a specific study and specific timelines, we can speak directionally to those. And so we're happy to take your questions, and we'll do our best to answer them as best as we can.
Thank you, Caleb, Yuri, and Dan. That was it is exciting. As you laymanize the words, Yuri, exciting is definitely apt. Now the first question, I've got two questions here that are related, so I'll ask them together. So what happens if there's too much protein produced? And the other one is, do you need to know the Syngap level, protein level in the patient prior to the trial? So I think they're kind of connected.
Yeah, so I can start with increasing Syngap too much is really not much of a concern for us. There hasn't been, I'd say one, just on our technology, the amount that regulatory RNAs control and are responsible for the expression of the genes that they regulate is within this physiological range of sort of like twofold or so. So even, in wild type animals, I showed that we got, a 1.5 to 1.7 fold increase. But in haploinsufficient state, which is what patients would be in, we really don't have evidence that we're going to go beyond twofold. So I think that's actually something nice compared if you look at there may be other modalities where it's more difficult to really control the extent of expression that you are looking to achieve. But for Syngap, with our mechanism, there really isn't. We haven't had any evidence to suggest that we're going to go beyond what would be the desired amount of Syngap expression. And I think to the point of, do you need to know the level of Syngap beforehand? I think it comes down to what's known about the genotype of the individual, but there's not really a way to measure Syngap directly. I think that's something us as well as others have thought about. Can you measure Syngap in CSF, for example? But that's how well that will work and will reflect what's in human brain is not clear. So I don't think knowing Syngap levels before an individual is on a trial would be required. I'll let Yuri maybe want to expand a little bit on the on the genotype aspect, but that's my thought.
Yeah, I think to Dan's point, well, it would be nice if we could measure people's levels more, just to help us with understanding what is the right dose amount. And I think that is something that's important for this approach, is that there are potentially the ability to be different doses. And so that will be one of the key goals of clinical trials, is to take studies that we're actually doing right now to help us determine what are the appropriate dose levels, and to prepare them for what are the dose levels that we would take into first in human studies. But with that said, we would not need to know. And so that would be what the baseline protein levels are. I think one thing I just want to underscore that Dan mentioned, because it is an important question around, could there be an overexpression that could be harmful in particular protein? There are other diseases where you see so-called like duplications that some may see, where having too much of a given protein can have a harmful effect. I think, as Dan mentioned, it's really important, this approach, what we've seen across many different diseases and across just the functioning of how these regulatory RNAs work, is that they work to keep things within this kind of healthy range. And so they don't go much above. And even when we try and force them to go much above, so Dan actually showed some data in the non-human primate, or in the monkey. And in that data, those monkeys actually had 2 copies of Syngap. So they already were at that like 100% level. And while we were able to push it up higher, and that gives us confidence that we can get the ASO to the appropriate parts of the brain, and that we're seeing increased protein expression, we didn't see any harmful effects of going a little bit higher. So I think that's the second kind of safety threshold for us on this question.
We've got lots of questions. I hope you're ready. So the first question that came in while you were talking, probably about IT, Yuri, was what sort of sedation will be administered?
Yes. Yeah. So it is quite common to have sedation. There can just be kind of general sedation, which is not true full anesthesia, but there also can be full anesthesia if it's required. So the neurology teams at most hospitals, and definitely at hospitals throughout Australia, would have a protocol for, depending on the needs of, say, your family member who might be affected with Syngap, what would be the safest approach to make sure that they're sedated appropriately, so that they don't move, so they don't harm themselves, and no one in the staff gets hurt. So that, yes, that's quite common.
Okay, Caleb, I might get you to stop sharing the slide, if that's okay. Is it? Yes. Perfect. A big Yuri, that's better. Okay, question was, would it be a lifelong treatment?
So I can maybe take that one. So the answer likely is yes. In ASOs, the benefit is that if we need to stop it or decrease the dose, we can wean it off. But what that would also mean is that in order to continue to see benefit, you would need to continue to dose. One of the things, if you look at other ASOs, is that many other ASOs that use similar chemistry, and I know we've referenced several times, so I'll continue to reference Spinraza, is that the dosing regimen for many of those is where patients will initially get a loading dose regimen. So they might get 1 or 2 loading doses, and then they move to a maintenance approach, and that might be then every 3 or 4 months. So it would be a less frequent approach. And then I think over time there are lots of efforts to understand what might be other ways of delivering. Those are not kind of ready prime time. But this would be something that would be a lifetime treatment, most likely.
And so just to reiterate that point, you mentioned there are teams that are working on other ways to administer ASOs without having to do a lumbar puncture, but they're not there yet.
That's right. There are a couple where look at ways where you could deliver IV, but there might be particular molecules that might help shuttle that across the blood-brain barrier in a safe way. And so what it's trying to balance is not getting ASO to the other organs that we want to avoid, or we don't want to have to give too much of a level. There are also some devices that have been used where you could actually put, say, a shunt or port into the system that has the CSF, so the lumbar spine and or the brain, and then you could shuttle the dosage that way.
Thank you. Now, there's a few questions around the risk of wearing out the good copy, the good Syngap gene. Is that something you have considered might be a problem?
I don't think that, these genes are active, throughout one's entire life. And I think it's the expression is regulated throughout life. So I don't think there would be a wearing out. Haven't really done an experiment to sort of show that, but we have done, repeat dosing, where we continue to see increased Syngap. That's on a time scale that's not, obviously throughout life, but we don't have any mechanistic reason to think that continuing to increase the expression of the gene for an extended period of time would wane over time. So we don't have evidence for that.
Yeah, and I might just add, I think it's an intriguing question. One, all of us, including family members with Syngap, already make Syngap. They just make 50% less. So that healthy allele is active, but it's really kind of functioning more as the blueprint, and then it's the machinery around it that actually take from the blueprint. So I think that's a way of maybe thinking about how this is not something that necessarily is going to get worn out.
I know you mentioned that you're still working through what the endpoints might be, but there is a question around epilepsy. So do you think? Yep, go take it, Yuri.
Yeah, no, great question. So, we're not going to be able to, at the moment, speak specifically to what a specific trial might look like. But with that said, I think for any trial, and you can look across other trials like in Dravet, in Lennox-Gastaut, for other developmental epileptic encephalopathies, the thinking is to measure multiple domains of disease. So obviously seizures and or epilepsy is one, but that is not the only one, because this approach is attempting to target the root cause of the disease. We want to look across not only potentially seizures, but also behavior, social, communication, motor, I think is an important one, gait, sleep, etc. So there will be numerous domains that we would look across in different stages of studies, depending on how long those studies are. Different domains of the condition may change at a different time or may take longer or shorter to look at. So some stages of disease may focus more on some particular symptoms than others, just because the time point may allow us to easier see change in that. I think the other thing that I just want to emphasize that Danielle mentioned at the beginning that's really important is this open label expanded access, which actually has multiple purposes. So one of the upfront purposes is for people who are in shorter studies, as well as if the study has a placebo, then those individuals could potentially roll into that open label expanded access where everyone could continue to get access to the potential medication. But it also allows us to continue to look at those domains of disease, like the ones I just mentioned, and look at, do they change over a longer period of time? So it actually continues to give us very important information about what might be benefit. And then lastly is on the safety side. It allows us to continue to monitor for safety, which is absolutely critical throughout this. We understand that living with Syngap is not risk-free. You all live with lots of risk every single day. But what we don't want to do is add to that risk. And so that's important for us to continue to create a database. And that database actually then goes to regulators when they look at this data.
Now, Josh has asked about anticonvulsants or other medications that his daughter Hannah takes regularly. What would happen in the trial if she participates? Would you stop them, or how would you wean off?
Great question. So, again, while I won't speak to what specifically the future CAMP4 trials might look like, in developmental epileptic encephalopathy trials, and this includes trials like in Dravet and others, typically the approach is that people enter the study with their existing anti-epileptics. So those are not stopped. What you try and do, though, is you want to make sure that there are not active changes being made. So typically what a trial protocol might say is something like, hey, the patient needs to be stable medication doses for some period of time before transitioning to adding that. So we just know what is potentially going to be contributing to anything that we see. What will oftentimes happen then is at some maybe later point, either in that study or even in the open label expanded access, there is an opportunity then, if let's say a person's or patient's seizures do decrease or are completely eliminated, then there may be a weaning protocol or withdrawal protocol where you may then start to drop some of those prior anti-epileptics that someone entered on. And so that's a pretty standard approach in other developmental epilepsies.
Now, there's a question from, thanks, Yuri. There's a question from Lani about the mouse model. She's asking the age of the mouse. That you saw amazing results in those humanized mouse. How old were those mouse, and then in equivalency?
Yeah, so these mice were, I would say, on the dosed as infants, and then sort of analyzed out for equivalent of maybe a 3 or 4-year-old. So they were on the younger side. We're thinking about doing experiments to look at intervening at different ages. I think it's good to point out some literature that has been published on this, where there was a group that actually performed a genetic trick and were able to reintroduce Syngap expression in older mice, and shown that even in mice that were adults, you could reverse a number of the different behaviors and seizure phenotypes and EEG and behaviors, even as adult mice. So I think that there's really a thought that, yes, there's sort of this developmental kind of wiring of neurons that is impacted early on, but there is a continued role for Syngap biologically throughout life. So intervening at different ages is presumed to have some impact. So these were younger mice. Something that I think part of the question was, did we look at seizures? I didn't show that data. This is something that we're looking into. I think part of the challenge there in these mice models, is that they don't have very overt sort of countable seizures, and they often require induction with a chemical. So it's a bit of an artifactual situation to generate these seizures. But that is something that we are exploring. But I think generally the fact that we're really reversing a number of other different phenotypes provides some confidence that anything that's dependent on Syngap should be addressable. So, but those studies are ongoing.
Thanks, Dan. I call you Dan because we're Aussie. I hope you don't mind.
That's what I go by. Yes, that's fair.
I'm also Dan, so you can reciprocate if you want. So there are some questions around the side effects. Side effects in any of the studies. I suppose there's a few questions on the non-human primates, but also on the mice. What were the worst side
Yeah.
effects that you saw, and also if you know of other side effects in other trials.
Yeah, I mean, I think there's, what I can say is that as part of, and I think I alluded to this in describing our program, uh, but for any target of interest, really safety and tolerability is really, we place a high premium on that because again, we can engage the target all we want, but if it's not safe, it's not going to be a drug. So we really need to make sure that the ASO candidates that we're looking at are well tolerated. So throughout the development, we always have an eye towards that. We always prioritize oligonucleotides that are well tolerated. And what that means is when we administer them ICV into the mice or IT into the non-human primates, that really there was no observations made that there was anything, anything of note happening. And that's the case with this oligo. We haven't seen any observations. That said, we do have to do the formal GLP toxicity studies that are required by the regulatory agencies before getting into humans, right? So that's where it's a more rigorous, focused, very elaborate exploration of the safety and tolerability, and that's like the next step. But to this point, even to the wild type monkeys where we're increasing expression beyond their normal level, there were no indications that there were no untoward effects that were observed.
Great. I wanted to ask about costs. So, if there's travel involved, who funds the travel, the cost to participate, and then in the open label access, is that also free access? So, just if you could talk to us a little bit about the costs.
Yeah, I'm glad you raised this question, Danielle. I think it's really important. So, one, I think it's well understood that participating in trials of any type, one, there's risk involved, but there's also just an enormous amount of time and effort that impacts not only the patient family member, but the entire family. And so, while the best way that that can be supported is by covering those costs. So most companies, and I would expect this to be no different in Syngap, would support and cover the financial cost of travel arrangements that would also include not just for the patient, but also oftentimes some reasonable number of family members to be there to support them. I do think it is important to emphasize that, particularly in an early stage study, the amount of travel and the timeline for participating in some of the different assessments can be significant. And so, so the amount of travel, the timeline spent is taken into account so that we can really support families, because the goal is to make sure that anyone who would want to participate in the trial and who qualifies to participate in the trial is able to participate in the trial, and that financial resources or other types of barriers can be removed. I think the other thing too is we, like many other groups who are sponsoring research, particularly in humans, try and do as much as we can to also minimize what requires travel and see what can be done at home. And not everything can be done at home, but we do understand. And I think we also, I know from talking to you all personally too, that traveling with a Syngapian is not easy. And that's probably a severe understatement. So, so we will do everything we can. And I'm constantly learning from others to see how we can best make this any study that we do as manageable as possible.
And as an extension to that, I mean, Australia is geographically huge.
Yep.
So would you be considering if multiple locations in Australia?
Yeah, great question as well. So the thinking in many trials is that the earlier trials tend to be smaller. And so what we want to balance is we want to make sure that the existing sites get consistency and standardization in practice, rather than like have many sites, but they only see or treat one or two participants in the trial. So oftentimes what that results in is that the earlier trials tend to have fewer sites, but as we get to later trials, there tend to be more sites, and sometimes you can have satellite sites. So while we can't give like the specific sites yet for future trials that may occur for Syngap, I would expect that something very similar to that would be the case. But I do want to underscore, I think, and particularly this is very relevant to Australia as well as it's relevant to many parts of the US, for example, is how we can manage what actually really requires a visit to a site versus what can be done at home, or what sometimes can even be done at satellite sites, like a standard lab or blood draw may not require transportation all the way to the original site. It might be able to be done at a local facility.
Brilliant. I've got a few more questions. Sorry.
Please.
We've got some interested people. So one of the questions, just because it's quick and easy for me to ask, is how do we express interest in participating?
I think it's amazing that people are expressing interest participating. That really warms my heart and others. You know, right now we don't have any particularly specific guidance, but I would say the fact that you're here at the appropriate time, when a particular Syngap clinical trial is being present, there will definitely be communication about that. I do think the other thing too is, share this with your clinicians, share this with your physicians, because for some physicians, they may have a lot of experience in trials, but others it may be new. And so helping you help educate them, just like you probably have about Syngap with your child, and that they're also learning. So I think you share that, and that those are other communities that we are also actively engaging with, is the clinical community as well.
Okay,
Yuri,
just a few more.
if you don't mind, I might just add, hopefully you all get the sense we want to be as open and transparent as we're developing this program as humanly possible. So Yuri hit the nail on the head. You're doing all the right things right now, and we will be, maintain our partnership with SRF, and we will be communicating, hopefully again in partnership with our SRF, once we've got some information about any trial activity that may be ongoing. So just stay connected is all I would say. Yeah.
We're keen is what is the message we want to relay back to you. Would the level of dosage change for different age groups? Is a question from Philippa.
Yeah, so right now, actually, one of the key things we're doing is we're actually doing a number of studies to look at different dosing in different animals. And then those doses are typically translated to what we call human equivalent doses. And so while we don't have specific dose levels figured out, it is not uncommon, particularly for younger individuals who might have smaller brains, to require smaller doses or concentrations than others. Typically the frequency, so like the loading dose and then maintenance dose, those typically would stay the same.
And that there is a question around frequency. So what would you expect the frequency to be?
Right. Without knowing ourselves yet exactly what that would be, I think the best model is that the chemistry that we've used for our ASO is essentially equivalent to the type of chemistry. And what I mean by that is like the backbone that holds all the base pairs together, or the nucleic acids, that is equivalent. And so we can use that to extrapolate what we anticipate the dosing frequency would be. And we're using an equivalent backbone as what has been used in other approved ASOs like Spinraza. So I think Spinraza is probably a good comparable or analog, where they have multiple loading doses, and then they have some maintenance period on a quarterly basis.
Okay, great. So a few more questions. Keto diet, I would assume, is the same category as the anti-epileptics, that you can stay on the keto but don't change your ratios.
Correct. Those are the types of things that oftentimes study teams they want to track, because we want to know just what people come in on. But usually a specific, particularly common diet that oftentimes individuals with developmental epilepsies have, those typically are just tracked.
Yeah, great. Would you expect if Jen's asking what happens if you need to be removed from the trial for whatever reason? Firstly, I'd like to know how would you be, like, why would you be removed from the trial? But secondary to that, what would then the expectation be as far as any progress that you've made? Would you regress back to preclinical?
This is an interesting question. I don't think we have a great sense of what this would look like. I think the important thing to point out, and Dan can feel free to jump in on this as well, is that the so-called half-life, so this is a measure of how long it takes in a given tissue to get to 50% of the levels, is quite long with ASOs in the brain. And this is actually a good thing because it does allow for that less frequent dosing. And so that means then the activity of the drug would occur. Now, the open label expanded access, as I mentioned, so if someone were to participate in an early trial and get one of the first doses, there might be some period of time where one has to monitor for safety before they can get doses in the open label expanded access. But the goal is to get those individuals, if they so wish, they would not necessarily be required to, but if they were so wish to enter the open label expanded access, to get them on that as relatively quickly as possible.
And is there a cost to access the ASO in the open label access?
In clinical trials, the entirety of the participation is covered by the sponsor, which in this case would be a biotech.
Yeah, and so even beyond the trial, that
Correct.
If you participate in the trial, you don't
in the open label expanded access.
Yeah,
that would also be
in that
covered
category.
well.
Yeah,
in that category. And any trial cost participation in the open label expanded access is thought of as just one of the other components of trial and study participation.
Yeah. Okay, just two more. What sort of medical support is offered if there are changes with seizures or behavior?
Not sure if I totally understand the question. I mean, the full participation of the study, and that would include like other components of medical costs that are associated with the study, would be covered. If there are sometimes things that fall out of that, then I think that would normally be under one's insurance. But I might need to, and happy to take any like offline questions if someone wants to follow up. Then yeah,
sure.
Better understand.
Yeah, that's Jade who's asked that. So Jade, perhaps we can get some more information from you and get that answered properly. Okay, last question. If seizures are not already present, could the trial cause them as a side effect?
I mean, all kinds of side effects are possible in all trials. I think it's just important to reiterate that adverse events, as they're oftentimes called in clinical trials, do happen. They can happen. And so that's why all the animal data that we follow, it is, I think, from a mechanistic perspective, thought to be unlikely that increasing Syngap levels towards normal would introduce seizures in someone who doesn't have them or worsen seizures. But it's hard for us to obviously speak with any certainty about what potential negative effects could occur. And everything is tracked. So everything is tracked so we can understand even if there might even be other causes to it. Is there causes of participation? Is there causes of travel? I mean, there are many cases where sometimes, and I know like traveling makes all of us tired, makes Syngapians tired. Being tired can increase your risk of seizures. So there are a lot of aspects of study participation as well as delivery of care that are important for us to track.
The other angle is the natural history of Syngap is that seizures can naturally develop over time. I mean, the average age is 2, but sometimes people can have seizures or notice seizures later. So that could be coincidental potentially.
Thank you for mentioning that, Danielle. And actually one other, I think, quick thing, because you mentioned natural history, is the natural history participation that that you and the community have is really, really important. That's been a huge benefit for us. Just like Caleb mentioned at the beginning, getting access to some of the cell lines has been critical for us. The work that you all have been doing to support research across early basic science research as well as natural history is absolutely critical, because it does help us understand what the natural course of disease is, so that we could observe an effect if if a medicine is able to do so.
And as an extension of that, Professor Ingrid Schaffer's got a natural history study that all Australians can be part of, and I'll share the details of that. And that's to help us get some good baseline data. Okay, we're going to close up in 30 seconds. Last one is, how many participants are you looking to kick off for the trials?
So while we're not yet able to give specific numbers for a potential CAMP4 clinical study, what I'll say is that early studies, so the kind of phase 1-2 that Danielle described at the beginning, those tend to have smaller numbers. And then as you move into later stage trials, those tend to have slightly larger numbers. Rare disease conditions like Syngap tend to have overall smaller numbers. So you might see studies in the sizes of early studies might be in the 20, 30, 40, 50 range. And then later studies might be in the 40, 50, sometimes even up to 100 range. I think the important thing too is that sponsors and clinicians can sometimes work to also do other types of studies in parallel to that, might be for special groups or special types of population, different age groups, etc. So there are many different aspects. And I think I shared this at GETA. I think it's important to consider is that there may be different stages of study that might be most appropriate for a given family member or a given patient. So just because there's an early stage study doesn't mean that might be the best or most appropriate fit for you. And just because there's a later stage study doesn't mean it might be most appropriate fit. So some patients and family members sometimes find that different stage studies are a better fit for them.
Wonderful. Thank you. I think we've answered all of the questions. I think we've got someone saying bring on the ASO trial in Australia. So as you can see, we're super keen. We want to thank you very much for waking up early for us, and we are really excited to hear about the next stage and updates as you go. So please keep us in mind.
Thank you so much, Danielle, and thank you to everyone for having us.
Yeah, thank you. This was great. We really appreciate it.
Yeah, thanks so much.
Thanks,
Appreciate
everyone.
the thoughtful questions. Yep.
All right.
Thanks.
Bye-bye.
Take care.
See you next time. Thanks.